viruses, as far as we get today, and we probably won't get that far. I mean, I told you to read ahead a little bit, but we're gonna pretty much go through, the size, some of the genetic makeup, and I hope to get through the lytic cycle, but we'll see how it goes. I don't think it's gonna be like a whole glutton of information right before the exam, and I know I used to hate that, I'm sure you do too, so try to keep it somewhat fair. Other questions? So essays, I gave some suggestions. Don't use the outline as, this is the only thing he'll ever ask me kind of thing. I mean, the outline does have a lot of stuff on it, but there's suggestions for essays. I've really tried to jump on a few themes that you can expect. So, I guess that's it on that. Okay, we're gonna attempt a review today. We'll see how that goes too. Like I said, I've been battling with my Advil in my pocket. I've been battling Blackboard most of the night, and we'll see what happens on that. But one thing that should work is our virus lecture. So let's think about it. For review, I'd like to have about 45 minutes for that. So we're out of class at 1:45. So how about lecture for a half hour and review? Okay. You need money? You say shut up if I or something somewhere. Wouldn't that be right? Half hour? Are you okay with that?
Sure.
Because there's other people that I know. That guy was at my time to shut up. Okay. Are we up? No. Open. So let's see, we made it to size, right? And we got into definition. And I think that's where we dropped it the other day. Am I right in recalling that? Okay, so we have these different terminology, the study of what a virus is, the virion, the particle itself, the virioid, which sounds like virus, which is not a virus. We have this term we call bacteriophage. Why they don't call them bacterioviruses, I don't know, but it's an industry term. So you also see the word phage. When you see that, think virus that infects bacteria. That's the deal there. And we'll talk a little bit about more about tempered phage, but it's here now. A virus whose genome is able to replicate along with the hosts. If you're kind of reading ahead, that is synonymous with the lysogenic phase of virus reproduction or replication, I think is a better way of saying it. If that means nothing to you, we will be covering that at some point. So, okay, this is where viruses, why they get so diverse and why they're also kind of hard to keep a handle on them. Essentially, viruses will either have RNA or DNA. They don't have both. You have both in your cells, as you well know. Viruses don't. So that's the first choice. So you're like, well, why would they have RNA, not DNA? But when you think about it, if you come into a cell with messenger already made or plus strand RNA, you're pretty good to go. But sometimes some of the mechanisms come in different ways, and it offers different advantages for these things, realizing they're probably working basically off a probability scenario, not any kind of adaptation scenario. So then with each type of nucleic acid, you have one or two choices. No virus has all of these. Okay, these are separate scenarios. So already we're up to 4 different ones. DNA, that's SS, single strand or double strand, single strand RNA or double strand RNA. Okay, we will look at that. We'll classify some of our viruses by these different scenarios and talk about what it means to have that type of situation. There are two situations also that are an extension of this. This is where you have retroviruses, okay, that have reverse transcriptase. So reverse transcriptase is like the big surprise of like the century. I mean, everything that's living follows the molecular dogma of DNA to RNA, then RNA to amino acid, so transcription and translation. Retroviruses can produce reverse transcriptase, an enzyme that goes backwards, so they can take RNA and make DNA. Okay, this really was a surprise to people because nothing else, that's living does that, and also is a big boon for the molecular biology market. The very fact that you can extract DNA is really cool. We've learned a lot of things about microbes and things that we can't see. But the question always is, well, you've pulled out the DNA out of that mud sample, out of that gut sample, out of whatever. How do you know it's really being expressed? How do you know that organism is even active? So people start pulling out messenger, which is really fragile and it's a little bit harder to work with. But they can go backwards and then transcribe or get the gene information by reverse transcriptase. And we have a variant on the double strand thing with the herpes virus. Right now we're just going to let that sit for here. Viral replication. I'm not going to play the video right now. We'll try to get to it. The reason I like to say replication, and the book says replication, is really viruses don't reproduce in the classical sense of that word. Bacteria reproduce, okay, they have binary fission, some have budding, we reproduce, okay, but that process is through meiosis, not mitosis. But there's some kind of exchange of genetic information, some kind of replication. If the viruses of genetic information that goes into reproduction, with viruses, their replication makes more numbers, but there's no mating that ever goes on. There's no real exchange. What happens with exchange with viruses is they sometimes cut up other DNA by accident, and they repackage it by kind of mistake. Okay, and we'll look at that. So viruses, we've talked about that. Most viruses are small. We've already covered that. We'll go on to the next part. This is just a table. I think it's good to look at now. We're going to cover a lot of these, so it's just a good reference table, but you can see that mainly we look at, we're interested in animals, although plants, tobacco mosaic virus was the first one to be really formally discovered. We started seeing the differences here and then the structure of the genome. But one thing we do see is that we're not talking, they're talking about nucleotides, we're not talking about a lot of genes with these things, okay? They basically come in, make more of their own nucleic acids, make coatings, and then work on trying to get out of the organism. The structure of the viruses are simple. We're going to start out with, there's two basic kinds. There's the naked virus and the envelope virus. Okay, so the naked virus is just this simple type of situation. You have the nucleic acid, just went through that, DNA, RNA, single strand, double strand. And then it's covered by a protein coat. So in some ways you go, oh, well, that's kind of like a cell. Remember, the animal and plant cells have a cell membrane and then oftentimes have a cell wall of some kind. This is a very simple structure. This whole structure is wrapped around the nucleic acid is called a capsid. Okay, and the capsomeres that they refer to here are like the individual little parts of the protein that are coming together to form this sheath around it. So together, there's more coming here. So together, this is the nucleic acid portion, the capsid portion, so it's called nucleocapsid. This is the simplest kind of virus. Okay, we're going to start building on that idea. And here is the envelope virus. You'll notice right away that we have the nucleocapsid in the center of this, and then we have this envelope. As it says there, the envelope is lipid, okay, and then some embedded proteins. These embedded proteins often are produced by the virus, although sometimes they can also be from the host cell, okay, which is clever. Because with your immune system, if you're looking for a virus, and that's what your immune system's doing a lot of the time. What cells are you, what cells are me, and what's something that I don't recognize? That's what it's doing all the time. You're teaching your immune system, okay, that one's okay, this one I want to knock out. And then you get a vaccine, and it makes antibodies and stimulates the immune system to, learn to attack, other things, and so we keep teaching our immune system how to do stuff. Well, if you're a virus and you're moving out of either the nucleus of an animal cell or the membrane, one of the last things they'll do through budding, a lot of times, is put that part of the envelope around themselves, and then they'll have these protein codes on the outside. And like I said, it's really variable, and it's hard with viruses to be, cover all the different variations, because that's what makes viruses so pathogenic. Year after year, there's a lot of adaptation and mutation that goes on with them. But so we got the formula for the basic types of viruses. This just gives a little bit better idea of the way things look. Ligands are the proteins that stick out. You might have seen that word before, biochemistry or something else. So how can we start putting viruses together? Which kind of shapes do they make? Okay. Here's the tobacco mosaic virus. Here, the rabies virus is more kind of a bullet-shaped looking thing. I'll get more pictures of that in a few days. But the simplest way to make a virus is to just make the capsomeres into a corkscrew and just kind of wind them up here in kind of a helical fashion and put it around the DNA or RNA we're talking about. This one, the icosahedron, we kind of think that this is one of those little, paper games you might make. Like, can you make a 20-sided polygon or something? And you think, well, it's just that artists don't know how to draw circles or something. It's not any of that. This is the shape of it, 20 sides. It turns out to be really stable and easy to make, and resist compression. So you'll see this icosahedral symmetry, and it's not a mistake. Okay, these protein spikes may be there, they may not be there. It really depends upon the type, I shouldn't say species. But you'll see this icosahedron. Kind of hard to see with that electron micrograph, but very, very efficient way to make stuff. Okay, now this is a naked virus, okay, still, but it's a naked complex virus. Okay, there's no, why am I saying that? Probably most of you have already figured it out. There's no membrane around here, no spikes going on. Really, if I take away this little lunar lander thing on the bottom of this phage, I have that icosahedron naked virus thing. So that's the nucleocapsid. This is a phage. It's looks like T4 phage, of course. And what we have here is this tail. We have these fibers, and there's even a plate on these things. Basically, they go to the. This is an E. coli phage, so it'll find the receptor, the right area on the E. coli. It'll lyse and inject the nucleic acid. They're double-stranded DNA viruses, and so it'll just kind of take kind of a squat. They've actually looked at this. It'll like compress and then the DNA will go into the cell like that, and then the DNA will, there's a sequence in viruses. They start out by producing more of their nucleic acid first, and then they'll incorporate some of the other nucleic acid in order to make capsomeres, and then the tail fibers and the base long tail fibers. And then it'll do self-assembly. It'll basically find the copies of the nucleic acid, assemble around that, and then create death to the host by creating a bursting size that will actually pop the viruses out of there. So, different virions can have enzymes for both infecting, for getting in or out. So basically, neuraminidase enzyme that cleaves glycosidic bonds, that makes sense to us. We know that we have sugar bonds all over on cell membranes. That's a good way to get in or out of the host cell. So the main point here is that viruses are specific. We've always known there's viruses around. People have been sick, and they started to figure out viruses a long time ago. But the extent of it is still becoming more in focus for us. For instance, we know there's viruses that we get, animal viruses, and those plant viruses. We just learned there's bacterioviruses. It came as a surprise to me, but I guess after I thought about it, it wasn't a big surprise, but there's archaea viruses. You think, why in the world would we never get an archaeal infection. It's just something that's independent from us. We get infections from bacteria, but there's archaea phages, archaephage, which I find very interesting. There's a whole different line of these little nucleic acid vectors moving things around. The other thing that is surprising to us, I think I told you part of the story, that when I was called up Kurt Sull after looking at viruses, and he said, "You idiot, you're not seeing the viruses directly," which is kind of what I meant, but he was very explicit about his response. But the number of viruses, we said there's about 1 million bacteria in a ml of water. A lot of you in lab proved that to yourselves last week, and told your friends and tweeted it across the country. They're amazing. But as amazing as that is, 10 to the 6th bacteria per mil of, we did Lake Springfield water. Fresh waters have about 10 to the 9th, about 1 billion viral particles per mil. You know, now, the first part you go, oh, so what? They're small. But I mean, on the other hand, so what? That's a lot of them, you know, kind of a thing, you know. And so this coming down that these things are specific is a big deal. But people are still trying to figure out what they do, okay? We know some of them. You go to the sewage treatment plant, you ever want to do this lab? We go and we get E. coli phage, and they're all over there. You know, you have lots of E. coli there. It's easy to find them. It's easy to count them. It's easy to make them. We think that some of the algal blooms may suddenly go away because of a viral infection. There's just a lot of stuff people don't really yet know about. But one thing we're pretty happy about is that, you know, all of them don't take on all of us all the time. Otherwise, we would not just simply be here. Okay. How could you count viruses through culturing? Okay. I just talked about, or made a reference to E. coli phage and how you can go to a sewage treatment and you get. Now that's, probably not the, it doesn't sound like an exciting Saturday night activity, but it is academically kind of interesting that you have viruses in such high numbers that they're basically killing off the bacteria in the very system that allows us to be relatively disease-free. Without sewage treatment, we would have a rough time of it. So one thing you can do is called the plaque assay, this word here, the plaque assay. And the way this is done, and if any of you want to do it, I find the viruses very interesting. We start with the host. In this case, we'll just make it E. coli. So we start with E. coli, and we grow up a nice culture that, and we go out someplace and get a sample. I've been talking about the sewage treatment plant, but if there's a certain virus infecting some kind of bacteria that you're interested in off of another source, this is just an example. Okay, so we have a viable host, and we go find our sample. What you do is you start out with a petri dish or some agar in the bottom of it. And then you have your culture, maybe a few hundred microliters, and you put in your virus. The best way to get a virus, or sewage treatment, or a sputum sample, whatever, is to filter. Okay, you filter out all the other stuff. Viruses are smaller than 0.2 microns. You know, we actually catch them on 0.02 micron filters. But anyway, you filter out everything else, and so you take your filtrate, you mix it up with the host, and you let it sit for a few minutes, usually around 20 minutes or so, because you have to allow the host to be infected. Okay, and you're hoping that you have more host cells than you do viruses for this to work. Okay, you can play with that, but for now, let's keep it simple. And so you mix your host with the viruses into a thin agar called top agar. Is it on here? Well, this is the top agar. Okay. And then you pour that thinner agar over your agar petri dish, and you put it in the incubator, go out, have a cup of coffee, email your mom, whatever it is you wish to do. Now, you come back once the host cells have grown, probably 18 hours or so. If you had no viruses, it'll be a confluent lawn of cells. It'll look nice and opaque and kind of foggy. You say, oh, all the E. coli grew, that's great. If you have E. coli phage, one of the many in there, they'll go and they'll go to a host, they'll infect it, they'll blow it open. Then they have a whole bunch of them all around that where they first infected. They'll go and infect the other ones, keep blowing cells apart. They don't swim, they don't have motility or anything. So if they do that a bunch of times, they eventually just kind of run out of being able to spread too much, and they make clearings into the top agar. The clearings are the place where they killed the host cells. So given that you had originally one phage infecting one cell, one could go back and visually try to estimate how many viruses you had. Okay. Again, it's culture dependent. It wouldn't work on, of course, on culturable bacteria, which is kind of the rub in things. Only 1% of things are cultured. Wow, there's a bunch of viruses and a bunch of things we're still not too sure of, but we could do this in these cases. So, and here's a picture of a plaque. Like I said, we've done this before. Anyone interested? I don't have anything scheduled, but I always love looking at viruses as much as the next guy. So, here's where I'd like to get, and I think I will by my time quota. I'd like you to know the steps for the basic virus replication cycle. Okay. Again, I try to distinguish between replication and reproduction. As a biologist, perhaps some of your friends would not, but that's what I think you should do. We're going to do this very simply with a host cell. You can think of this as E. coli. It could be an animal cell, but this most looks like, once we learn more, like a phage kind of situation. So we start out with attachment, and sometimes that deals with adsorption. We'll find out they're using both those terms because attachment usually happens with bacteria. Adsorption is where a virus will actually be engulfed by the host cell. Some of the animal cells will do that. But either way, we got the virus, the virion, the virus particle, coming in contact with a host. Okay, and for host, insert bacteria, animal, plant. Okay, then entry. So if upon absorption you already have that, that's fine. It could be injection, like we talked about, T4, basically, and then the outside stays on the outside. With animal viruses, sometimes, like I say, it's engulfed and comes in, so those two steps are a little bit more or less indistinguishable. But anyway, you got to get that DNA or RNA into the host cell, so that's penetration. Okay, Now we're moving into the next step, synthesis. In this case, you have then the nucleic acids first of the virus, nucleic acids being reproduced, okay? And then the viral proteins, sometimes there'll have to be some changes between the viral genome. If you have antisense or non-sense RNA, you have to make sense RNA. Do you know what I'm talking about? Does that make sense to you that I'm talking about sense and plus and minus strands in RNA? Who doesn't know what that is? Oh, so most of you do. You want to explain?
No.
Okay.
I do not.
Okay, you know when you have double-stranded DNA, you have two sides of DNA. They're anti-parallel, that kind of thing. And a gene is transcribed off of one side. That makes positive messenger. So from positive messenger, whichever side of the DNA that is, you can go directly into translation. So positive is messenger that can go directly into making protein. If you have the opposite side of the DNA that could make positive strand, but it can't be transcribed directly, or if you have RNA that has to be made into positive strand, that's the negative strand, or sometimes called nonsense. Sense, nonsense, or plus and negative. Okay. So basically, the easiest way, the easiest way is if you have plus strand RNA, that's active messenger, that's ready to go. So anyway, that may be happening in here, maybe a little bit of rearrangement. Then you have assembly, often just through auto-assembly. There's not like there's enzymes that come in and scaffold these things. And then you have release. Release can be caused, it can be induced by different factors. It can come from having a burst size, that is, getting so many viruses it puts pressure on the cell membrane. That can happen. As we saw, there can also sometimes be enzymes involved. We're being very general right now, like when we talk about all these different things. Let's see. Okay, attachment of the host, receptors, specific, and attachment of the virus to the host cell results in a change to both virus and cell surface that facilitates penetration. In some cases, if you're infected, the cell membrane will change and you don't get secondarily infected, which can be seen as perhaps a benefit in some host cells and some bacteria. Okay, this is the what I was talking about, the sequence of events. Okay, so we have time, okay, on here, and we have relative virus counts. Okay, so here we're adding viruses to a host. All right, this latent period. In this case, it's just talking about where you don't see virus particles because you've had then attachment and penetration. The early enzymes come out in this case. If there are some, certainly the nucleic acids proceed, then the production of the protein coat, that's the last thing you need. The early enzymes sometimes set this up, sometimes you can just go straight into this. Okay. Protein coat. And then when you have maturation, okay, then that's the assembly, and finally your virus numbers here go up because you've assembled viruses. In a way, this case, the viruses are prophage. That you couldn't recognize them unless you somehow pulled the DNA out and sequenced them. So they call that an eclipse period. I think that's it on that. Yeah, this recaps that, but we can stop here. What's important so far in the viruses is you studied in size, that genomic thing, having those four main differences, the fifth one being the reverse transcriptase case, the retroviruses, the size, okay, the relative shapes, morphology, and then the lytic cycle. Right now, we're going to cover one of those so far. We'll come back to the lysogenic cycle and getting an idea that the timing is really important, okay, which is not everything happens at once. Okay. Those are the main factors as we kind of enter into this whole introduction of virology. Any questions? This one is right on the thing. I wish I had that stuff. I'd be so much better at what I do. Okay. So, we're going to try to review things, and if it doesn't work, we'll go with something verbal, but I'm trying to make this somewhat engaging, so let's see what happens. Oh, hang on. Do we know if the sound works here? Does anyone know how that works? Well, we'll find out, I guess. We're going to try to do microbial Jeopardy. Do you guys, do you know this show? Do they still have it? I haven't seen TV in years. When I was a kid, my grandma would watch this, and we'd all, that's the end, you try to see how many answers you get. Well, this is what I'd like to do with us today. I thought, if nothing else, I'd get us thinking about questions. And if other things came up, you say, well, what about this, that, and the other thing? I could try to explain